All right, well, I just wanted to start off by thanking the Suddath family and the friends of Dr. Bud Suddath for making this opportunity available. When I look at the plaque with all the names listed on it, and I see all the students in the past who have had this opportunity to receive the Suddath Award, it just amazes me how much this has been able to help us. For me, I was able to go to a really great conference, the Genetic Society of America conference, and it was at Princeton University. I got to see the university, and I had a chance to present my research, and it was just a wonderful opportunity. So I'm really thankful to the Suddath family for making that possible. So here's a few pictures from the conference. It was held in August, so they thought that would be really clever and have the Olympic symbols with the yeast, the budding yeast at the top. So that was really bright. I had to show you that. And here was one of the beautiful rooms we had the conference in, and one of the theaters there. And we had a lot of workshops at night that we went to, and also poster sessions. So we got to meet some of the top leaders in the field and hear them present their research and present ourselves. And it was just a great experience and great for my CV as well. So I was really thankful for this chance. So today I'll tell you about some of the research that I've been doing in Dr. Chernoff's lab, and this deals with protein misfolding. So this is the key word here is protein misfolding. So within our bodies, there are thousands of different types of proteins that have to fold into perfect three-dimensional structures in order to keep us free of disease. But in some rare cases, a particular type of protein can just sort of go haywire and fold into some strange pattern, something like you might see if you just imagined it, something like this. And this can lead to neurodegeneration and also different diseases. And so the type of misfolding that I'm talking about today is the misfolding to form this fold called amyloid. So amyloid is like a generic term that many proteins can adopt this type of fold. And this is characterized by self-assembled, fibrous, cross-beta sheet-rich protein aggregates. So in humans, there are more than 40 different examples of diseases, and just a few of them are shown here, in which a particular protein, shown here in green, can start folding wrong into this amyloid form, and then it can cause diseases. And so you might have heard a lot about Alzheimer's disease in the news in the last 2 weeks. And they were trying to predict that in 2050, they think that the rates will quadruple to possibly even as many as 16 million people in the US having Alzheimer's disease. So these diseases are caused by protein misfolding, but there are no really sure ways to prevent these diseases, and there's no cure for these diseases. Although they can be treated, but usually the treatments are to try to alleviate the symptoms, and they don't really deal with the protein misfolding. So there's a special class of amyloid, and these are the ones shown here at the bottom. The transmissible spongiform encephalopathies, also called Prion diseases. And these are a special class of amyloid because this is called self-propagating amyloid. So it's kind of like the whole amyloid process is converting new proteins to make them fold in the same way that it folds. And so this is why it's a special class. Although it has been shown for most of these diseases here that to some extent these are infectious. They might not be infectious from person to person, and there are a lot of arguments about whether they could be or whether they couldn't be. But at least at the cell-to-cell level or the protein level, we can see that these can be infectious. So that means that studying these prion diseases can give us information into how these diseases might spread through different parts of the brain and how they might be infectious between people, possibly through like blood transfusions. So here's our prion model. So we have our protein that's misfolding, shown here by a box. This is showing that it has a different structure. It's shaped like a box. And here this prion aggregate can come in contact with non-prion protein, and this would only happen for proteins with identical sequence of amino acids or similar sequences. So these non-prion proteins can interact with this prion aggregate, and then they can use this prion aggregate as a structural template that they can use to adopt the same structure as the one that they encounter. They can then become part of this aggregate, making it larger, and eventually this can then become fragmented into more of these seeds here so they can interact with more non-prion protein, and the cycle can continue. And this is to give you more detail about the amyloid structure here, and this is to emphasize that not the whole part of the protein itself does not have to completely misfold. So here we see one protein molecule being made up by the green box and also by the yellow loop. So here we see the portion that's misfolded is shown here in green, and the portion that's still soluble that can possibly even function in the cell is shown here in yellow. And you see here how the prion domains here can then interact with each other and make the aggregate get larger. And this is the axis of the fiber that can form. And so once these proteins can start to aggregate like this, we can see that like for Alzheimer's disease, one of the peptides that aggregates is called A beta, and these can form plaques as they start to aggregate together outside of the cell. And then there's another protein in Alzheimer's disease called tau that can aggregate inside the cell in these types of tangles that can form. And these aggregates can then affect the functioning of the neurons and cause the death of the neurons. So in the Chernoff lab, we actually use yeast to study protein misfolding. And people ask us why we use yeast instead of other eukaryotic model organisms. And these are studied. Mice, rats, chimpanzees, and cows and other organisms are used. But we find that yeast are very attractive for several reasons. One is that they can exist as either haploids or as diploids. So they can have one copy or two copies of the genetic material. And so that can be really useful when you start knocking out genes, because you can have a haploid strain with only one copy and knock the gene out and easily see the effects of that loss of that gene, which you can't see that so easily in mice. You can make these disruptions, but it's much more difficult. And also, once you make these deletions in the chromosomes in yeast, it's really easy to make them, and then they can also be stored in the freezer at -80 degrees. So years later, you can pull them out of the freezer, you can have each gene deleted one by one, and you can do these screens through the whole library of all the deletion collections and see which particular genes might be affecting whatever phenotype you're looking at. Also, with yeast, we can get much faster results. Sometimes in just 2 or 3 weeks, we can have a result. Whereas if you worked with something like a cow, it might take 10 years to get results because of long incubation periods. Mice might take 6 months or longer, and other animals could take even more than 10 years. So it's nice to have that results coming really fast in. And also, yeast are they can be much cheaper to work with and easier to culture than other mammalian models. And there can be an argument that yeast could be safer, potentially, because there'd be less transmission possibly from a yeast to human, especially if you're dealing with yeast proteins that are not found in humans. But in some cases, we do use mammalian proteins in yeast, but we think there's probably less of a chance that they could infect humans if they're in yeast rather than if they're in other organisms. So one idea that we're interested in is a potential therapy that might could be developed. And this is a type of interference with prion propagation. So you saw the prion model, how it keeps propagating and propagating the same protein. But in this type of idea, here we have the protein. This is like several proteins stacked on top of each other as they're aggregating. The boxes show the parts of the protein that are misfolded. And here we see that we can use an interfering heterologous sequence that's slightly different. And it would actually be much shorter than this, but I just drew this to show you an example of how it would work. So portions of this sequence could be exactly the same as what you would find here in this protein molecule, but other regions could have some slight differences. And we think this would try to join onto the aggregate, but somehow it can inhibit other of the wild-type proteins from joining on. Somehow it can interfere with that. And it might be a way to slow or to stop progression or propagation of the prion. Another thing that we're interested in looking at in our yeast model is the prion species barrier. So here we have an example of what a species barrier is in mammals. So we have sheep, cows, and humans, all mammals and all able to produce a protein called PRP. This is the same one that caused Creutzfeldt-Jakob's disease in humans and Kuru, which is a disease of cannibals in humans. And in sheep, it can cause, when it's misfolded, it can cause a disease called scrapie. And in cows, it can cause mad cow disease. And here we show that in parentheses, these numbers here, we show how similar the protein is in the amino acid sequence between each species. And the arrows show where the transmission of the prion can go from one species to another, and this one is blocked here with the X across it. So in some cases, like an infected sheep that has scrapie, the proteins from the sheep could infect a cow if it came in contact with those proteins, if it ate them somehow in its feed. And it caused disease in the cow. A person could get disease from eating infected beef, but for hundreds of years, it's never been shown that a human could get disease directly from sheep. There seems to be a barrier here that prevents transmission of the prion state from this species to humans. So we're interested in seeing whether it's the protein itself, whether it's regions in the protein that have divergence, and trying to find out what regions could be causing this barrier, or also it could be cellular factors or a combination of all of these things. So we're trying to find out which things are responsible for preventing the transmission from one species to another. So in yeast, we actually use a protein called sub-35 to study the protein misfolding. And people ask us why we choose this particular protein, because a lot of different proteins in yeast can misfold. But one of the reasons why we choose this one is because we can tell easily when it's in the prion state and when it's not in the prion state. So it makes it a really attractive protein to study. And we study this using 3 different yeast species: Saccharomyces cerevisiae, Saccharomyces paradoxus, and Saccharomyces biennis. So we show that they're all very similar in this protein in the same domains. The end domain is the most important one to us. It's the prion domain, which is important for forming the prion. The M domain influences the solubility of the protein, and the C domain is the part of the protein that is essential for the cells to have because it helps to terminate translation at the stop codon, so that they die without the C region here. And the way we can tell if our sub-35 is in the prion form is to use an AD114 reporter allele and also to use color differences on YPD growth media. So here, when our prion, our sub-35 is in the prion form, it's called Psi plus in the prion form and Psi minus when it's not in the prion form. So here on the left part of the figure, we see the sub-35 not in the prion form, and here we see it is in the prion form on the right. So when our protein is in a soluble form and not misfolded, when it can help to terminate translation here at a premature stop codon, located somewhere in the middle of the gene for AD1, and this is called the AD114 allele. So when it comes to this premature stop codon, it's able to help terminate translation, and the complete AD1 protein cannot be made without having the prion there. So the cells cannot grow on media lacking adenine, and they're also a red color on YPD. But if we do have the aggregation of the sub-35, then we see that this doesn't function very well to help terminate translation. And so the ribosome just ignores the premature stop codon and completes making the entire protein. So that means the cells can make AD1 and they can grow in media lacking adenine. And also you see that they're a lighter color on YPD, more of a white color here. And so one of the ways we can use to examine the effects of heterologous poisoning is to use this technique here. So we just call it heterologous poisoning. So here we take a yeast strain that's deleted for sub-35 in the chromosome, and we put a copy of it on a plasmid here in the cell. This is this green loop that you see here. And this version is from Saccharomyces cerevisiae. So it's the top yeast species that you saw on the list of the 3. And we verify that this is definitely a prion, so it's Psi plus here at the beginning. Then we transform our strain with another version of sub-35. So here we have 2 copies of sub-35 at the same time. So this new copy can be identical to the first one. It could be from a different species like PARADOXUS or Biennis, or it could actually be a chimeric version where we mix and match both different regions from 2 or 3 different proteins. It could be anything we want here. And then we look to see, we let it stay in the cell for 6 days, and we look to see whether the presence of this new protein for just a short amount of time was able to destabilize the prion transmission of the SC protein right here, to see if it stopped the propagation or not. So we just look for loss of the prion phenotype. So we look at the end of the number of colonies that have the prion and those that do not have the prion. And you just have to keep in mind here that all that we look at here are all the same protein we originally started with. So we've already removed this protein at the end. We're just counting how many of these proteins can actually keep the prion. So when we use 2 copies that were identical to each other, both from Cerevisiae here, we saw all white colonies. So this shows that we did not have any poisoning because white means that it's the prion phenotype. So no poisoning when both copies were identical. When we switched out we put the new version in with the Paradoxus prion domain, we see lots of red colonies here in the picture. So that means that the original protein is now inefficiently propagating the prion. So it's had some loss of the prion. So we call this poisoning because we're actually interfering with the propagation. So we're poisoning the propagation of the prion. And so this shows you a negative control. So we introduced just an empty plasmid without any gene on it at all. We didn't see any poisoning here. This is when we introduced the same version that we started with. So we had 2 copies exactly the same from Cerevisiae. We essentially had no poisoning here. Here's when we introduced the Paradoxus version. We had 62% of our cells were Psi minus, or our colonies were Psi minus. So this is the same figures I just showed you on the previous slide, and I'm just adding a few more to this. So we also checked some controls where we took the first third of the protein prion domain from Cerevisiae here, and then the second and third portions we took from Paradoxus. And here we had the inverse of that, where we took the first third from Paradoxus and the other 2/3 from Cerevisiae. We saw this certain regions are more important than others for the poisoning. And here we also checked the importance of single amino acids. So So we looked at the sequences for the CEREVISIAE and the sequences for the PARADOXUS versions of SUB35 in the N domain. And we found a few key amino acids that were different. So we tried changing one and didn't see any results at all. We tried changing this one, and we did see that when we switched it, just this one amino acid, it actually removed the poisoning by switching that one from the whole thing being PARADOXUS to the whole thing being PARADOXUS, except for this one being more like CEREVISIAE at this point. So then we tried to do the inverse of this by actually putting this mutation in to the CEREVISIAE version, making it more like PARADOXUS, that one particular amino acid. And we did not see that this caused poisoning, so we decided to try a chimeric version right here, so it's more like PARADOXUS, but at this one position we switched out the amino acid and we found that this did cause poisoning again. So we conclude that a single amino acid residue is crucial for the poisoning phenotype that we observe. But one thing we noticed was that we tried the same poisoning experiments both in PARADOXUS and also in the CEREVISIAE species. And we used the same prion in both of these. So we took the prion from CEREVISIAE, we transfected it into this PARADOXUS strain, so they have the same prion in them. We verified that both prions were strong, and then we did the poisoning experiments. And we saw it here in this species, but not in the CEREVISIAE species. So we conclude that in this case, poisoning depends on the cell environment. There must be chaperones and other factors in the cell that must be influencing this poisoning besides just the protein itself. So we're trying to identify those factors right now. So in addition to this poisoning we've been looking at, we also did another technique that was very similar called a plasmid shuffle. So here we started the same as we did with the other experiment. This is the one we already looked at here where it branched off to this point. And here, instead of removing the new protein after 6 days, or the new gene after 6 days, we kept the new version of the gene and removed the old version. So we just swapped these out. So here we're swapping from CEREVISIAE to a new version right here. And then we're seeing if the prion state can be transmitted from the original protein to the new protein here, and to see whether it could efficiently get this, the prion or not. So we're looking at it like a species barrier type experiment. And we record how many of colonies are Psi plus and how many are Psi minus. And so this is just a simple version showing basically what we did. We did a lot more to this, but this is showing you the basic results. So here we started with CEREVISIAE protein. We switched it out both in the PARADOXUS cell and also in the CEREVISIAE cell. So these are shown here in parallel. And when we switched it from CEREVISIAE to CEREVISIAE as a control, we had high rates of transmission. This is exactly what we expected. When we switched this from CEREVISIAE to PARADOXUS, we actually saw still high rates, but not quite as high as to the same protein. When we switched this from CEREVISIAE to Bioengineering, we saw a huge reduction in the efficiency of the transfer of the prion from the CEREVISIAE species of protein to the Bioengineering protein. And this happened in both species. So we concluded that the prion transmission specificities are the same, both in CEREVISIAE and PARADOXUS, and that the species barrier parameters are determined by the protein's origin rather than by the cell's origin. This is different than what we saw for poisoning. And there's one more thing I want to tell you about, and this is about different strains in yeast that we found and how our strains could change from one type to another. So a strain, I need to tell you what a definition is of a strain. A protein of one and the same sequence, the same amino acid sequence, can initially, when it starts to misfold at the first time, it can form several different types of folds. So once it folds wrong, then when it starts to give this prion phenotype to the same protein to have the identical protein, whatever fold it originally adopted will be faithfully reproduced over and over again, as long as it's using the same identical protein. So we see that mammalian prion strains, they're called strains in the way they differ in their folds. They can show differences in the regions of the brain affected by the disease. They can show differences in incubation periods. So some kinds can be worse than others, causing early onset of symptoms. Some kinds can wait till much later in life before they show symptoms. They can show different severity of diseases and different rates and types of progression of the disease. So in yeast, you can also see prion strains too, and in yeast these are called variants, just to keep the confusion down because we already talk about yeast strains all the time. So they're called variants. So we can see differences in variants on different growths on -add media. So here we see a strong prion growing much stronger on -add. A weaker variant will grow much weaker. And here we can see color differences with the strong variants being more white color or more pink, and the weaker ones being more of a dark pink or reddish color. So here, this is showing our shuffle experiments where we started with a CEREVISIAE version of the protein right here at the beginning. We then switched from CEREVISIAE to CEREVISIAE in a shuffle experiment, and then went back in another shuffle experiment to CEREVISIAE again for control. So here we go from CEREVISIAE to CEREVISIAE to CEREVISIAE, and we see the same type of prion formed from YPD and from minus add growth. If we go from CEREVISIAE and then shuffle to PARADOXUS, we see a weaker phenotype, and then we go from PARADOXUS back to the original protein that we started with, and we still have the same type of prion. So we think it's, the actual fold of the protein has not changed here, because we're still able to reproduce the same type of fold in the original protein. But then we go here from CEREVISIAE to Bioengineering and then back to CEREVISIAE. So we have the same protein here at the end that we had at the beginning, and we actually have a different type of phenotype. It's like a weaker phenotype. It grows a little bit less on minus add. It has a darker color on YPD. And we tried to verify this biochemically by spinning our proteins down the centrifuge, the ultracentrifuge. When you see a strong prion, you see that virtually almost all of the protein will be in the pellet form here. The amyloid protein will be in the pellet form. Very little will be in the supernatant. If you have a psi- strain, virtually all the protein is in the supernatant and there's none in the pellet. And the weaker versions can have more in the pellet, but also some in the cytoplasm, or in the supernatant right here. And so this is showing you the strong prion isolate we began with almost all of it in the pellet, like we see here. And then when we took these samples here, 2 different isolates from this and ran them on a gel, we see there's a good deal of protein in the supernatant, which shows that we now have a weaker phenotype for our prion. So when we passaged from CEREVISIAE through Bioengineering to CEREVISIAE, it actually changed the type of fold that we have in our prion. So our overall conclusions from this research is that a divergent protein can poison a prion, leading to its loss. Amyloid poisoning by a heterologous protein depends on the cell environment. Variation at the single amino acid position can make a crucial impact on prion poisoning. The specificity of prion transmission is determined by the protein itself rather than the cell environment, and this is different than what we saw for the poisoning experiments. And the sequence divergence determines whether a prion variant will be faithfully reproduced or whether a variant switch will occur. And these are some of the people that have helped us with the species barrier project. And I'd like to thank the Suddath family one more time. So, thank you very much.